DNA synthesis and PCR. DNA structure was discovered by these two persons, Watson and Crick, and it is suggested that DNA is a double helical structures. These two DNA strands is binding to each other through Watson and Crick base pairing, and because of this phosphate linkage groups, DNA is a highly negatively charged polymer. So to neutralize these charges, DNA is bind to positively charged protein, so-called histone. So through this, the complex, the charge is neutralized. DNA, in the beginning, there was a simple double strand, but by binding to the histone, it forms a nucleosome and make further and further more complicated complex structure, supercoiled structure, eventually to make chromosomes. The building block of DNA synthesis is triphosphate molecules. As in ATP, the biological energy molecule, triphosphate contains a high energy which can provide the chemical energy to make a new bond. And the synthesis happens from 5' to 3' directions, and 3' OH group attack this phosphate, alpha phosphate, and by removing this pyrophosphate, new phosphate bond can be formed. For the DNA replications, first of all, the topoisomerase need to release the strain from supercoiled structure. And then helicase open up double strand into single strand template. From this side, the synthesis can happen from 5' to 3' and it can go smoothly and continuously. Problem is in the other strand. The synthetic direction is from right to left. The problem is it cannot be continuously happening because of this opening up and synthetic direction is the opposite. That's why there should be some fragment and then it could be connected later. And this is the predicted by the scientist named the Okazaki. That's why this fragment was named as Okazaki fragment. Mimicking the natural DNA synthesis, the in vitro setup for the DNA the replication method, so-called polymerase chain reaction, PCR, has been invented. The beauty of this work is it's very simple, repetitive the process to make many copies of a DNA. For this reaction, you just need thermal cycler and also building blocks of the DNA synthesis and DNA polymerase and primer. So everything should be mixed together in one mixture, and just by changing temperature, this reaction can be repeated automatically. The first step is the denaturation. Now, the target DNA, double-stranded DNA, can be separated into individual single strand by simple heating up to 95 degrees. And then the primer needs to be attached to the template. And this process can happen only at low temperature. That's why annealing process to lower down the temperature down to 60 degrees. Then the primer can attach to each point. Then the extension can happen by DNA polymerase, and the synthesis can be completed. After this reaction is done, the next step is also increasing the temperature to divide this to the double strand into a single strand again. And then this reaction can be repeated. The PCR reactions can be recycled by these temperature changes. The first step is increasing the temperature up to 95 degrees to divide this double strand into a single strand, and then annealing process to allow the primer attachment, and then enzyme reactions. And then this temperature cycle can be repeated over and over to make the amplifying DNA process. Through these PCR reactions, if the cycle number is n times, then the product number is 2 to the n. That's exponentially fast growing of those implication speed. Here the key catalyst is a polymerase. Polymerase is a protein enzyme. Then in this reaction process, how can the polymerase can survive the high temperature up to 95 degrees? Well, the secret came from bacteria living in hot spring. It's a really interesting idea. Well, the bacteria which lives in hot spring, they should have the high stability at high temperature. So the scientists try to isolate the DNA polymerase from this bacteria and apply that into PCR technology. And with this, the technology development, Kerry Mullis, he got Nobel Prize in 1993. Here is one application of a PCR. So during the COVID-19 period, so we had to test the biological sample, which may contain very small amount of virus particle, but those small amount of virus particles genetic information can be amplified by PCR, and it can give us the final the call whether we have the infection or not. So as you can see here, this PCR technique has been applied to many different fields and has a very high impact in biological systems.